ways in these two conditions, aerobic and anaerobic. Anaerobic means without oxygen, right? So here this before this point, right? This -6, -4, -2. So these points are considered during anaerobic conditions, right? And after this +2, +4, +6 are of aerobic conditions with oxygen, right? So what is the time interval they measured the expression? At what time intervals have they measured the expression? Huh?
2 hours.
2 hours. Here you can see. See, after 2 hours, again, after another 2 hours, expression is measured. That means the mRNA is measured, right? Another 2 hours, expression is measured. Yeah, also before 2 hours, again, before 2 hours, expression is measured. So we can see that how the genes are producing mRNA, right? What we can see is that what can you say about the expression levels of these genes? What can you say about the mRNA production, or what can you say about the behavior of these three genes during aerobic and anaerobic environments? Are they behaving in the same way? Or all three behaving in the same way?
No.
No. Then what can you say about the three?
The last gene behaves in the same way.
Yeah, that means not changing,
Yes.
changing its action. No? The production of mRNA, it's that gene, whether it is aerobic or anaerobic, the producing the mRNA at the same level. That means the action is at the same level. This green one is it increasing or reducing? First gene?
It's increasing.
Increase and after some point reduce. Yeah? Yeah, 10 and 5. This one is reducing. Yeah, yeah. Let's say I think. Yeah, this is expression level of gene at checkpoint J, right? This checkpoint. Here you can see after plotting the expression levels. Here this green gene expression level goes up till 10, here till 10, and goes down after that. And this red gene, right, it goes down, further down. This is not affected. We can say the action of this gene is not affected by the oxygen, whether it is anaerobic or aerobic. Okay, any questions here? Let's see what is. Here I think, yeah. So normally these expression levels are converted to logarithms, log values of base 2 to compare, right? So So these are the log values. So after taking, normally you convert into log values and then compare. Switching to logarithms of expression levels. Now in here, some more genes are considered. Here you can see this actually this yeast has, I can't remember correctly, but 480, yeah, something genes, 21,000. Yeah, we'll see. I think the exact number maybe here, right? So you can see here, some more genes are considered here, and the expression levels are noted. So this we call as gene expression matrix. You know what is meant by a matrix, right? So if you have done R programming, you have worked with matrices. And also in MATLAB also you will work with matrices. So this is a gene expression matrix, and these are the levels of gene expressions measured for each gene, right? And this is the plot of the different expression levels. Here you can see some expression levels are go down, and this is again for 1, 2, 3, 4, 5, 6, 7 points. Expression level measured at 7 checkpoints, right? So some, and the other thing is that you can see that some genes, right, are working in the same manner. Here you can see 3 genes here working in the same manner. No? They go up here and more or less same. And these genes also working in the same manner. And these genes also, these two working in the same manner. So that we can say some genes working together, right? So one advantage of getting this gene expression, learning or tracking this gene expression is to see, identify the genes that are working in a similar manner, right? So we know that how many genes do we have, human? One, two, 30, 40, hundreds, millions, billions.
Thousands of genes, 30,000. 30,000.
Yeah, it is around, you can say around 25,000, right? Yeah. So we know that when we have around 25,000, so when we have the gene expression matrix for 25,000 genes, right? We can't identify the, say when we have 25,000 records. So we have in rows here, we have the genes, right? So when we have 25,000 records, it is not easy to see by plotting all the genes like this. So there we do the clustering. No, we cluster similar so that similar genes will be clustered together. So there we can identify the genes that are behaving in the similar manner. Here with this, one of these we call as gene expression vector, right? So gene expression, we know that vector means vector in our MATLAB, we talk about vectors, that is like 1D array. This is an array, right? You can say 1D array. So you can see here the gene names are in different colors, so that the expression levels also identified. The genes that are working in the same manner are colored by one particular color. That means the red ones. These are behaving in the same manner. Here. So these are the number of genes that yeast has, right? 6,400 yeast genes. Yeah, so the yeast gene expression matrix is done in 1997 by Joseph. He has measured the expression of 6,400 yeast genes of 7 checkpoints before and after the diagnostic shift, right? So that you have 6,400 this way, 6,400 rows, right? So you have 6,400 rows and 7, that means 7 conditions. We can say 7 conditions. So the condition or environment will change with time here, right? The oxygen, in terms of oxygen level. So this, if we do this matrix for the whole yeast genome, we will get 6,400 into 7 gene expression matrix. So the goal here is to the goal of this person, Joseph, in 1997. So the goal of his work was to partition all these genes into clusters so that genes in the same cluster have similar behavior, right? Genes in different clusters have different behaviors. So I think you all also have worked with clustering, right? I am referring to the section Miss Amali did. So you also try to cluster. So when you do the clustering, right, you will end up with the genes that have similar behavior in one cluster. So likewise, you will get several clusters of genes. Right? So when we cluster, so if we have clustered correctly, we know that within the cluster, you have the genes that are very similar in behavior. No? But if you take a gene from 2 clusters, one gene from one from each, right? 2 clusters, they will be different. I think you have learned 2 good properties of clusters. No? What are the properties of good clusters? 2 properties. You have heard the word cohesion, no? In software engineering, cohesion. In modules, say we say within the module, no, the code, we talk about cohesion. Or we say when we have several clusters, So when we have several clusters, when you have several clusters, right? We know that clusters means groups, right? Right. So we have genes in here, right? We have another set of genes here and another set of genes here. So as I said, as we show in the plots, right, these genes will behave in a different manner and these genes will behave in different manner in producing the mRNA or in making the protein, right? So some will will at the beginning they will produce more protein and will reduce after some time. Some will, if you can remember the plots, right? Some will say decrease. Yeah? So some genes behavior will be like this, will decrease, but some genes they will not be affected. This set of genes we can say not affected. Yeah? They will have the same level of expression. And we can say this set of genes, they may say their expression levels. So that means say at first 2 hours, they will produce this much of mRNA, and at the next 2 hours they will produce this much of mRNA, and next 2 hours this much of mRNA, and we can say in the anaerobic situation, aerobic, it will transfer from anaerobic to aerobic, right? So when it's in the next hour, we can say oxygen is available. So it will produce the very less. That means its mRNA production level will be dropped drastically, right, at the next hour. So likewise, we can identify sets of genes that are behaving in a similar manner, or genes we can identify clusters of genes. Any questions here? Anything you didn't get? So hope everybody understood. So you will see more practical applications, right, in the next slides. Now, let's see what else is there. Yes. Okay. So in here, you can see that genes as points in multidimensional space. Now, in here, we have, though we show it in here, like in 2D space, right? Those genes have to be present in multidimensional space, right? That is because we know that N into M, this matrix, right, gene expression matrix, so that N points in, we can say it is N points in M dimensional space, right? So that we can say that it is a high dimensional data. So this is here. This is a more practical example of gene expression data, measuring gene expression data. In here, you can see the gene expression is measured in colon cancer patients. Colon is a part of the digestive system, the very end of the digestive system, colon. You know that some people get cancer in this part of the digestive system. Right? So in 1999, this Yuri alone measured expression of 2000 genes from 40 colon tumor patients and 40 healthy people. Now you can see the two conditions here. The two conditions are here, the patients and the healthy people. Okay, patients and the healthy people. Now, if you can remember in our earlier yeast example, 7 conditions were there, right? That is 7 conditions in the sense that they measured the expression at 7 different time points. So at each time point, the conditions change, right? So in here, the 2 conditions are patients and healthy people. Normally, this data taken, actually this expression taken from the same patient. Though I don't know why maybe here they have taken from 40 healthy people, because that we know that when you take one person, he has the cancer cells and normal cells both in his body. Yeah. Normally you take, you know that there is a database, huge database, TCGA. I think I showed you last time, no, when we discussed about that CBio portal. Yeah, so in those data sets, data for healthy people also available. So healthy, they have taken from the same patient from a healthy cell. Right? Because you know that cancer is not on the whole body, you know, in some cells in the body. So you can take expression in the cancer cells and the normal cells. So in here, this here says maybe we in 1999 it might have done in this way, right? So 2000 genes from 40 colon tumor patients and 40 healthy people. So you know that we have 2000 genes, but in here on the expression is measured in only 2000 genes. So this matrix we can say for the patients, right? So can you tell me what are in rows and what are in columns? Rows and columns. In this matrix, what are the rows and what are the columns? What is in columns and what is in rows? I mean, the type of data, right?
2000 genes are in the rows and healthy and the unhealthy are in the columns.
Yeah, okay, yes. So yes, the columns, you have patients or samples, and rows, you have genes, right? So in here, they say that this is a 2000 into 40 gene expression matrix, right? That means that this can be for a 2000 into 40 means yeah, but if it is this is like that means that two here it is said two this kind of matrix. No? That means one matrix is for cancer expression data and the other matrix can be for healthy. Right? Or actually you can combine those two because the rows are the genes are same. And you can see here the numbers are same. The samples, they have taken same number of samples. Here, the healthy also 40 and cancer patients are 40. Yes. Okay, let's move on and see what else. So the goal here was to find genes with significantly different expression vectors. We said that expression vector is a thing like this. No? To find genes with significantly different expression vectors in tumor patients as compared to healthy people. That means we can use those genes as potential cancer biomarkers. So what are cancer biomarkers? Cancer biomarkers means genes that can be used to mark the cancers, biomark the cancers, or genes that can be used to identify the patients with colon cancer. Okay, so how it is going to happen? By finding the genes with significantly different expression vectors in tumor patients compared to healthy people. So imagine that this gene, right? So if this vector is significantly different from the healthy people, right? From the healthy people. So if the behavior of this gene is significantly different in this in all the cancer patients, right? You know that this is these all are these are not time series data, right? So this say in the same patient in the all are patient, right? So if it is you can see here all are negative here, right? But imagine in the all expression values are negative for this particular gene. Why negative means? What do you mean by negative? Means the expression of this is low. The expression of this gene is low, right? So if the control, right, the expression of this is positive, that means there's something different, something that we should see, right? So in that case, you can use this. If it is significantly different, we can use this genes as cancer biomarkers, colon cancer biomarkers. Or what you can do, say if someone is suffering from cancer-like disease, right? By extracting this gene, that means by monitoring the expression of this gene, right, of that patient, we can say, because we know how it is behaving now, right? We know now how the behavior of it in healthy person and cancer patient. So we can see the expression level and decide whether it is a colon cancer or not. Right. So that is how these gene expression matrices, gene expression levels, help in developing cancer biomarkers, and how you can use those to identify cancer patients. Actually, you can say, like, not only the patient is having a cancer, you can tell the type. You can say it is a colon cancer, it is a colon tumor. Because in case of breast cancer or any other cancer, the genes that will be altered will be different. So that for those, there is a different biomarker. That means that you will be looking at different sets of genes. Different set of genes will be altered in that in those genes, so that you by getting the gene expression values, you can tell the type of the cancer. Okay, any questions here?
Madam, I will change the dimensionalities in that previous example, so that dimensionalities change.
Dimensionality in the sense that you mean that we said there are many dimensionalities when there are so many genes, like, no?
Yeah.
Is that the one?
If there are different dimensionalities, and why we check for why we changing the dimensionalities? Is there any sense in changing dimensionalities?
That is because we can't work with those. Actually, it is called as curse of dimensionality. Curse of dimensionality means when the dimensionality increases, like when there are so many data points in different planes, right? And some methods will not work. So actually we call it as curse of dimensionality. So actually our next lesson is to, so that as some of the methods are not going to work, we need to reduce the dimensionality. Reducing dimensionality means reducing the number of genes or features. In here also, actually, we are reducing the dimensionality. Reducing the dimensionality in the sense here that to test for colon cancer, right? We don't need to check all the genes, check the expression of all the genes. For example, imagine a patient comes to the clinic, right? So that we need to check whether that patient has a colon cancer, right? So that we have this cancer biomarkers developed, right? So that we know that, imagine we have to check only 10 genes. We have identified 10 genes that will differ from the normal person, right? So that we check only 10 genes. But if we have not developed these biomarkers, or if we have not reduced the number of genes that we have to check for, we have to study the whole whole genome expression or whole gene. I mean, all 25,000 genes we will have to study, right? But we have identified the genes that are significantly different and identified the cancer biomarkers so that we can easily see by studying only 10. So when the number of genes high, the dimensionality increases, like you, so that you have so many data points, right? And you can't work with those because those are in so many different planes, right? So actually, that is called as curse of dimensionality. So you will see in the next lesson that we need to reduce the dimensionality, right? And we see the ways to reduce dimensionality. And why do we need to reduce the dimensionality? And it is actually a whole area of study. High dimensionality. Last time, Amali, Miss did her project on this set of cancer data obtained from TCGA or CBio portal, right? So that they are it is the data available for patients 20,000 to 25,000 genes, right? So we can't work with that much of data points, right? Because sometimes the signal is covered by the noise, right? So we have to reduce the signal to noise ratio as well. So we need to identify some significantly different genes. So that that is actually very say whole area of in machine learning, right? Reducing the dimensionality. Okay. Okay, so that's good question. So moving on. Here, in here you can see here that if you say if you plot it like this, no, but these data points can be in different planes, right? So that you watch this video, right? I have added this to the LMS, right? So that actually here this speaker mentions about the this m-dimensional space. So in here, this gene expression matrix is you can see the plot here, right? So these are the different clusters, right? So this is for the healthy person. So you identify clusters of genes, right? These genes behave similar. These are in similar manner. These are in another way. So if you try to plot the cancer data, right, on the same plot, so we expect, right? We expect if these genes behavior is same in the cancer patients also. So you expect the values to be same. That means it will plot on the same spot, right? But this is somewhat same. You can see here, somewhat similar. So here you can see the more or less same. But These genes here, these two groups are very different, right? So that they are significantly different in their behavior. So this is another cancer biomarker, talking about another cancer biomarker. This mama print is maybe from mammogram, no, mama print is a test that evaluates the likelihood of breast cancer recurrence. Breast cancer recurrence means we know that the appearing again, the cancer. So you remove the cancer cells, and sometimes after some time, some cells will grow again, right? So this is a test that evaluates the likelihood of breast cancer recurrence based on expression of just 70 genes. So by studying the expression of 70 genes, right? By studying expression of 70 genes, they actually this is a commercial service, right? They will not tell us the 70 genes that they measure, right? They may be that is their secret, but they will. You can pay the money and you can get the test output. So we also actually trying to build a biomarker for diabetes. It is said MODY. MODY means maturity onset diabetes of young, right? So that is the diabetes type of diabetes found in children, school children, right? So that is also a genetic disease, right? So we also we do the study the expression and sequencing all that in the National Hospital, right? The doctor, they are endocrinologists, you know, who is working with the diabetes, right? Because he takes all the data and do the sequencing and measure the expressions of the patients that are coming into into that clinic at the National Hospital, right? So we are going to get the data, and so our task is also to develop maturity onset diabetes in young. So our task is to identify the MODY type of diabetes. So they are all. We also have to identify the 70 genes. Sorry, some genes, right? Definitely should be less than 70. I think 70 is too much, no? I don't know when this, but maybe between this. This is not just the occurrence or presence of cancer, no? This is just to see the likelihood of breast cancer recurrence, whether it will come again, no? Yeah, whether the patient will get it again after the surgery or after all the treatments, right? So that for that MODY, I said that diabetes type, there is no genetic test available. There is one in the in West, right? But you know that in Asia, the disease is different. Like our people, the genetic changes that we get and the gene changes that we get are different from the West. So that we can't adapt their test with our patients, right? So that we have to develop this cancer biomarker. So that's it about this video. Any questions? If no questions, we can go to the PowerPoint. So this video, to those who, I didn't record this because you should, because those who are not here should can listen to the video, right? This video has audio so that one can listen to that rather than listening to me, while there is audio here. Okay. So let's get back here. I think you all can see. Okay. So this is about finding differentially expressed genes. Now you know what is meant by differentially expressed genes. Right? Differentially expressed genes means genes that are of which the expression is different in different conditions. Give me, I will come back in 2 minutes, right? You also can have some water. Okay. Now, as I said, in here we are going to talk um, finding differentially expressed genes, because finding differentially expressed genes are important in developing useful biomarkers, and also to study differentially expressing genes, right, in plants and all this, to like it will help in case of plants. We can say if you can identify the genes, imagine genes that are important in increasing the yield, crop yield, right? Aswanda Median genes. So the environments, if you can identify the environments that will help in increasing the expression of genes that are important in getting more harvest. No? So that in a way that is important, or in controlling disease conditions, or in many ways it is important to identify, important to study about differential gene expression. So to start with, as we said, we have to in studying gene expression data, we have to work with high dimensional data sets. High dimensional means, we know high dimensional means so many rows, but less columns. For example, if you take our genome expression data, high dimensional data means we have this way. This way we have, if we take rows, columns, we will say large number of columns, variables, features. So we can say this way the columns. So this actually will we can't refer by the row columns, because we can represent in different ways, right? So if we can say, do it like this, I would say like this. I would say number of rows less, and now there are so many columns. This kind of data set, columns, or we call this as features. So features here in human gene expression data, human or any gene expression data, we say features as genes. So that means if we store features in columns, we have, in case of human genome expression data, we have 25,000 genes, right? And rows, we can say we can't take 25,000 patients, no? Say maximum, we can say we can have 200 patients, right? So that you can see that this is not symmetrical, right? This is 25,000 and this is 200. So this is the nature of high dimensional data. So here it says which are data sets having a large number of columns, variables, or features, or genes. In this data set, features refers to genes, right? So that is what we call as a high dimensional data set. It is it is valuable to select features. So we can't, as I said, we can't work with this high dimensional data set, right? We need to select features. We need to select set of genes to work with, features or genes, right? So as we can't work with 25,000 genes, we need to select genes. Does anyone having question?
Ma'am, so
Yes.
in the previous example in the video,
Uh-huh.
In the data set, you said that the rows will be having the genes and
Yeah, that can be. So in that sense, yeah, that can be. In that sense, we will represent the data like this, data set like this, no? In that sense also it will be like, so we can say in here, we can say it is 200 into 25,000. Yeah? So in here we can say 25,000 rows and 200 columns.
Okay.
No? Actually, normally in the gene expression matrix, no? In the matrix you have rows or genes. This, it is like this. No? If you can remember, in the gene expression matrix, the genes are in rows, and in columns you get the samples or the patients, right? If you can remember, in the yeast example, you had different time points in the columns. Still, you had only 7. No? If I can remember, can you remember the number of genes in the yeast genome? It will say 600.
6,400.
6,400, yes. So in the, if you can remember the gene expression matrix, it was 6,400 into 7. No? So this is high dimensional because these are the features, right? I think some of you, I think all of you have done machine learning, right? So in the machine learning, we talk about features. So in here also, I am referring to the same concept, right? Features. So that here actually our features are not like the width, height, not those, yeah? Color, or we are not those. Our features are genes, right? Okay. So hope you are okay, right? Okay. So so as I said, we can't work with this higher dimensional data set, right? We need to select some features to work with because it is valuable to select features as to reduce the size of a data set being studied to make its analysis more efficient and even feasible in some cases. Because it is important to reduce the size of the data set so that it is easy to analyze the data set, right? so that means analysis will be more efficient because it is easy to work with less number of features and even feasible in some cases. Make data analytics more effective. That means, as I said, the signal to noise ratio, right?
Recording
We
in
need
progress.
Yeah, so the signal to noise ratio will affect the analysis. So it is important to have only the few important features. So this is particularly important for bioinformatics data, computational biology data, because as we are most often involved in high dimensional data sets. This is curves of dimensionality. Refers to various phenomena that arise when analyzing and organizing data in high-dimensional spaces that do not occur in low-dimensional settings, such as the three-dimensional physical space of everyday experience. Right? So this is obtained from Wikipedia. So that that is why we need to reduce the dimensionality, or we say to reduce the number of features. Feature selection is also called as dimensionality reduction. Right? Feature selection also called as dimensionality reduction and contributes to data reduction in the end. So it is very important to biomedical in biomedical data. It is very important in biomedical data due to an often large number of features available. The curse of dimensionality, example, number of gene expressions, in number of gene expressions. So there are many feature selection methods exist. How we can do the feature selection? One thing is that we can reduce the redundant features. You know that you don't have to, if you take a data set, right? If you consider a data set with features like age, date of birth, you know that when you take a clinical data set, in this TCGA data set, there are clinical data sets as well, like for those cancer patients. In the CBio portal, if you browse through the data, you will find there's clinical data also available. So there, if you see the age and date of birth, both are there, you don't need, no, that is redundancy of data, you can drop one. And also there can be some features, irrelevant features, like patient ID may not be important. Experiment, sometimes there can be some data that is not important for our analysis. Also, features may impede the data analysis method from performing well. Like when you have many features, overfitting can happen. No? Overfitting means that it will overfit to the data that you build the model, no? To the data that you build the model, but it will not be a good generalized solution. You can't generalize it to another data set. Right? This is about feature selection. A minimal set of relevant features for future tasks, such as I said, for diagnosis treatments. That is what we call as feature selection, selecting a minimal set of relevant features for diagnosis of diseases, treatments, etc. Gene selection for future prediction. When you select the genes, right? For example, in a training set, gene 1, 2, 3, 4. Here you can see in experiment 1, this is how gene 1 to 4 behaves. in experiment 2, this is how it behaves. Experiment 3, this is how it behaves. And experiment 4, how it behaves. So when a test sample is given, right? So we have to decide at which condition. Like this we can say different disease conditions. Now, we know that cancer also will be in different stages. Now, at the very say primary stage, or at intermediate stage, or at the latter stage, so that accordingly that stage actually you can identify from the genes, from the gene expression, right? The genes, from the genes that are expressed. If you look at the gene expression data of a particular cancer at different stages, the expression levels are different. Expression, the genes, the behavior of the genes are different. So that when a test sample is given, we can say this is can be in E1, like E1 or E2, let's say, maybe more close to E1. Yeah? So there will be, so your model will try to identify it correctly when you build the model. Right? So these two are, so the E1 means this is a normal patient. Right? So these two normal. Okay, so that is how we use, that is how we can use the gene expression, and also how to identify a particular condition, particular sample using few genes when you have identified few features, few relevant features. So how many minutes do we have? We have 10 minutes. So within that 10 minutes, I would like to, I think we can finish this. Yeah, this one. So the in between parts, we will discuss next day. So feature selection for gene expression data, I think this part is covered by Kamal, is covered. Differential gene expression aims at determining whether different genes are expressed in patients of different medical conditions or between patients and normal subjects. So mainly we are looking at whether different genes are expressed in patients of different medical conditions. Right? We know that, I think you looked at this array, right? In here you will get these cells in 3 different colors. Some can be black, right? So these genes are underexpressed. If the gene is in green, we say underexpressed. This is intermediate, and this is overexpressed. That means this gene has produced a lot of mRNA, means it has overexpressed. If you can remember, I think you can see this. So this sample can be from the control. This sample can be from the patient, right? So these are the, we can say these are the mRNA they have produced, right? So you color these two in 2 different colors. This you color with green, that means the control sample. And this you color with red, that means the patient sample. And then you put both into this tray, right? So they will, these can be genes. So mRNA sequences of genes, they will go and bind to with the same spot, same gene. No, that because that we know that for that they will go and bind with the complementary strand, right? So at some point, some gene can be there and for the same gene, maybe expressed here and expressed here as well. Now in the control and the patient. In that case, those spots will become yellow. No? If it is expressed, overexpressed in here, the patients, so this will go and bind more. This gene will be more in patient, so that it will be, this spot will be more red, so that we can say this gene has overexpressed in this patient, in the patient. So these color codes can differ. No? Normally, green is used to the control, right? But in here we can say this is control and this is control and the patient samples, or it can be say, as I said, it can be from 2 stages of a particular disease. Any questions here? I think you are familiar with this. Okay, let's move on. So this is that mRNA array. You can see here some spots are red. That means this gene, red means it is overexpressed in the disease. This gene. Okay. So these are some methods for selecting differentially expressed genes. These are some R packages, right? You can use these R packages also to select differentially expressed genes. So then next, the visualization. We know that heat maps are used in visualizing gene expression. These are some of the other visualization methods. This is the heat map, right? This you can see here. This way it is the genes, and in here the samples. So that we can say this gene is overexpressed in this sample, this patient. Okay. So you know that this heat maps, right? Using heat maps, we can show the clusters. You know that here these genes or these patients here, right? These patients have behaved somewhat similar, no? Because in here you can see these genes, in their genes, these genes, right, are underexpressed in all this number of patients. So this gene expression and using the heat maps and clustering, right? So you can use the clustering and identify and mark clusters here. Heat maps allow you to mark the clusters. You can cluster genes or you can cluster patients. If you cluster patients, you can identify subtypes of the disease. You can say this subset of patients, here they are, here we can see a common behavior in these genes in this patient. We can say this subset of the patients have a particular subtype of this disease. So that can be a very good finding, right? So that you will have to treat them differently, right? And also to identify them also you can use these genes, this subset of this subtype of diseases. So this gene expressions, clustering, and heat maps. Heat map is just to visualize, allows you to identify subtypes of the diseases and then to treat the subtypes differently. Right, so that we will, any questions? Questions? I have some questions. Can you quickly answer this question, question 1? Quickly add your answers to the chat. We have only 3 minutes, 1 minute per question. Can we have the answer for the question 1 in the chat box? Is it difficult? Okay, one answer. 3. Okay, so it says, I don't know whether the question is not clear. In finding differentially expressed genes, we work with normal population only. No, you can't, you need 2 population. You can't, there is a, so A is not an answer. By having only the normal population, you can't compare 2 disease populations. That is correct. Using 2 disease populations, you can disease in the sense that infected or with the diseases. You can compare there, because you can say that you can take the breast cancer and colon cancer patients to see whether there is a difference between the gene expression. 1 disease population, this is also can be used, because you can see, as I said, say a population of cancer patients, right? And you can check their say gene expression, say at the beginning and at the end, to see whether there are sorry, at the end means after, you know that when you take a cancer, there are stages. At the stage 1, 2, 3, 4, like, right, at different stages. You can check the expression values. And will there be any meaning to you if you use two normal populations? No. So the answer can be these two. Any objections? Okay, next one. Heat map can detect the underexpressed genes only. Each square shows expression of the genome. Patients or conditions shown in columns. Genes shown in rows. So, answer for the question 2. Question 2, quickly.
Recording stopped.
Heat map. Heat map is the one that we looked at, like the visualization one. How to visualize gene expression data. Can detect under expression genes only? No, you can detect the overexpressed genes also. And each square shows the expression of the genome. No, each square shows the expression of one gene. Patients or conditions shown in columns. Here we have the screen. Okay. Here we can see the here you can see this. So the columns you have patients, correct? And genes, rows, you have genes shown in rows. So we can say these two are correct. Na? Any objections? Next one. Each square in heat map shows gene expression levels. Correct, right? Each square in the heat map. Where is heat map? Here each square shows that each square shows the gene expression values of each gene. Okay, then we'll continue this lesson next week, and also we have to discuss about the prediction. That means how to say when you have developed the biomarker to diagnose the diseases, that means when you have identified some genes to diagnose the disease, how to predict whether that particular person is having disease or the type of disease, right? How to do that we will learn in the next lesson. And also I will be adding the R code to do this dimension reduction and to identify the set of genes and to draw this heat map, right? I will be adding the code, and that code actually that is a Python notebook. I found it in one of the Coursera courses. I think you are familiar with the Coursera courses. So that I will be adding that link. So those who don't have registered in Coursera courses will have to register, and you can access the notebook there, right? So all the code there, what you have to do is just to play a little and see, and the other thing is you will not be asked to write the code for the exam, but you should be able to understand the code, right? Like you will be given the code and asked, but you will not be asked to write the code, because we write the code with tools, no? So the tool help is there for us always. So you don't have to write the code on the right. You should not do this. You should. It is not expected you all to write code without a tool. That means on the paper, right? Okay, then let's meet next Tuesday at 1 p.m.